If you take any living thing and zoom in far enough, the smooth, continuous surfaces disappear. Instead, you find a microscopic geometric grid. Every visible organism is constructed from these discrete, invisible blocks. They are the baseline structural and functional units of all life, cells. This drawing shows how that underlying grid was first documented in 1665. Robert Hooke was examining thin slices of dead cork under early magnification. Looking at this rigid lattice, Hooke thought the empty structures looked like the small, Spartan rooms of a monastery. He named them cells. Hooke was looking at dead tissue. It took another 9 years and the refinement of brass microscopic tools with better lenses to see what was actually moving around in the water. In 1674, Antony van Leeuwenhoek focused his equipment on a drop of pond water and became the first person to document living cells in motion. Over the next century and a half, scientists like Matthias Schleiden and Theodor Schwann compiled these isolated microscopic observations into a unifying framework known as cell theory. But the theory had a mechanical gap regarding where these units actually came from until Rudolf Virchow provided a critical update. Virchow summarized his findings with a strict Latin axiom, omnis cellula a cellula. All cells arise strictly from pre-existing cells. Because a cell can only come from another cell, the structures inside it have to be capable of sustaining chemical reactions and copying themselves perfectly. To see how they manage that, we need to map the architecture inside the boundary. This diagram will represent a generalized eukaryotic cell structure. We are going to build a composite model, combining elements found in both plant and animal cells to establish a structural baseline. For plants, the absolute outermost layer is the cell wall. It is a rigid, cellulose-based barrier that provides intense structural support. Just inside that wall, and acting as the primary outer boundary for animal cells, is the plasma membrane. This membrane operates as a selectively permeable phospholipid bilayer. It actively controls exactly which molecules are allowed to enter or exit the system. Past the membrane, the entire interior volume is filled with cytoplasm. This jelly-like medium suspends the internal structures and acts as the staging ground for the cell's metabolic reactions. Deep in the center of the cytoplasm sits the primary controller, the nucleus. The nucleus has its own distinct anatomy. It is protected by a nuclear envelope, houses a dense central nucleolus, and contains fibrous strands of chromatin. That chromatin stores the DNA. It holds the complete genetic blueprint for the organism, establishing the nucleus as the absolute command center. The heavy outer walls and selective membranes exist primarily to maintain a highly stable chemical environment, ensuring this central command unit can dictate complex operations without external interference. Moving immediately outward from the nucleus, we find a dense network of folded structures handling the physical production of cellular materials. This schematic highlights the endoplasmic reticulum, or ER. It forms a maze of membranes physically connected to the nuclear envelope. The section studded with small dots is the rough ER. Those dots are ribosomes. Their presence makes the rough ER the primary manufacturing site for synthesizing proteins. The outer sections lack these ribosomes, giving them a smooth appearance. This is the smooth ER. Without ribosomes, the smooth ER handles a different chemical workload, synthesizing lipids and detoxifying harmful byproducts. Once materials are synthesized, they are routed to the Golgi apparatus, an isolated stack of membrane folds sitting further out in the cytoplasm. The Golgi acts as the chemical processing and shipping center. It receives raw proteins and lipids from the ER, modifies them, packages them into vesicles, and secretes them to their final destinations. Together, the nucleus, the ER, and the Golgi form a continuous, highly coordinated industrial supply chain operating strictly within the cytoplasm. Running a continuous manufacturing line requires an immense amount of power. This structure is the mitochondria. Note the highly folded inner membrane, which drastically increases the surface area for chemical reactions. This organelle generates that power through ATP synthesis, functioning as the primary energetic engine for the entire cell. To manage the inevitable waste from these processes, the cell utilizes lysosomes. They contain potent digestive enzymes to break down debris. Because rupturing them would digest the cell itself, they are often referred to as suicide bags. For general storage of water, sap, and waste, cells use vacuoles. In plant cells, the central vacuole is massive, often taking up a majority of the internal volume. Finally, plant cells contain an exclusive category of organelles called plastids. There are 3 specific types: green chloroplasts to capture solar energy for photosynthesis, colored chromoplasts for pigmentation, and leucoplasts strictly for storing starches and oils. The architectural layout we've just mapped out directly mirrors the foundational diagrams used in biological studies, illustrating a complete system of internal specialized compartments. Eukaryotic life survives entirely through this strict internal architecture, dividing the heavy labor of survival among highly specialized organelles. This multi-compartment factory model applies exclusively to eukaryotes, the biological domain containing all plants and animals. There is an alternative, older biological architecture, the prokaryotic cell, represented primarily by bacteria. This table provides a structural comparison of these 2 domains. Starting with the command center, eukaryotes have a defined nucleus enclosed in a membrane. Prokaryotes lack a nuclear membrane entirely. Their genetic material gathers in an undefined nucleoid. Looking at the internal organelles reveals the defining architectural threshold. Eukaryotes are packed with membrane-bound organelles like the ER and mitochondria. In prokaryotes, membrane-bound organelles are entirely absent. Without these internal membranes, a prokaryotic cell cannot separate its chemical processes into isolated compartments. All metabolic reactions share the same internal space. This lack of internal infrastructure limits their scale. Prokaryotes measure roughly 1 to 10 micrometers across, remarkably smaller than eukaryotes, which scale up to 100 micrometers. Their manufacturing equipment differs too, relying on 70S ribosomes rather than the 80S type. Look closely at the far right column. The doodles perfectly illustrate the contrast, the blank interior of the prokaryote versus the dense, compartmentalized interior of the eukaryote. The presence of internal, membrane-bound compartments is the absolute foundational dividing line in biological classification. It dictates how a cell scales, how it manages energy, and what forms of life it can build. This brings us back to Virchow's opening axiom: all cells arise from pre-existing cells. For biological continuity to hold true, a cell cannot simply snap in half. It must systematically duplicate its entire internal architecture: the outer walls, the membranes, the energy engines, and the exact genetic instructions. The cell functions as a self-contained, fully operational microscopic universe, continuously duplicating its entire architecture to sustain the larger organism.